Have you ever wondered how doctors figure out what's wrong with a patient? How they diagnose, understand, and then, hopefully, treat a disease? Well, if you have, you are absolutely in the right place. Today, we're going to pull back the curtain on the field that is, quite literally, the foundation of modern medicine: pathology. To really set the stage, let's start with a powerful quote. This comes from Sir William Osler, who's basically a rock star in the history of medicine. And what he said really nails it. Pathology isn't just another subject you learn, it is the language of disease. It's the key to everything.
So what is pathology exactly? Well, the name itself kind of gives it away. It's from two Greek words, pathos, which means suffering, and logos, which means study. So it's the study of suffering. But more than that, think of it as the crucial bridge that connects all the basic science you learn in a lab with what a doctor actually sees in a real live patient. It's all about understanding the nitty-gritty of how diseases change our bodies, both in structure and in function. To really get a handle on any disease, I don't care if it's the flu or a rare cancer, a pathologist is always asking three fundamental questions. It's kind of like they're the ultimate detective, but the crime scene is the human body.
And this right here, this is a perfect way to visualize that framework. You see the patient at the very center of it all. And pathology is this process of figuring out what's gone wrong by asking about the causes, the mechanisms, and of course, the consequences. Let's break those down. So first up is the why. This is what we call etiology. What is the actual root cause of the problem? Is it something genetic that you were born with, or is it something you acquired along the way, like an infection, an injury, maybe something in the environment? This is always square one of the investigation.
Next, we ask how. And this is pathogenesis. This is the plot of the disease, the step-by-step story of how that initial cause, that why, actually creates the illness. So how does a virus actually make your nose run? How does one tiny genetic mistake lead to a massive tumor? Pathogenesis maps out that entire fascinating journey.
And finally, we get to the what. What are the consequences? This is where the rubber meets the road. It includes morphology, the actual structural changes in our cells and tissues that we can see, and of course the clinical significance. What symptoms is the patient feeling? What signs can a doctor see? All of this leads to the diagnosis, the prognosis, and finally a treatment plan.
Okay, so how did we even get to a point where we could ask these deep questions? Well, let's take a quick trip back in time to see how our understanding evolved. It's a really cool story of how we went from superstition to pure science. It's wild to think that in prehistoric times, getting sick was seen as a curse from God or maybe the evil eye. Then came Hippocrates, who started to focus on rational observation. Later, Morgani started connecting a patient's symptoms with what he found during an autopsy. But the real absolute game changer was Rudolf Virchow in 1858. His cellular theory was a massive leap. For the first time, people understood that disease wasn't some external demon. It was a malfunction of our very own cells. Illness came from within.
It's funny. Even the symbols we see all the time in medicine, like this one, the caduceus, have these incredibly deep historical roots. The snakes were seen as having healing powers because they shed their skin, a powerful symbol of regeneration that has stuck around for thousands of years.
Now, what's so cool here is you can literally see the focus getting sharper and sharper through history. Hippocrates looked at the whole patient. Morgani zoomed in on the organs. He actually started the Clinical Pathological Conferences, or CPCs, where doctors get together and review tough cases, something they still do today. Then Virchow zoomed in even further to the individual cell. And then Paul Ehrlich gave us the chemical stains to see what was happening inside those cells. Each one stood on the shoulders of the giant before them, taking us deeper and deeper.
But of course, the story doesn't end there. Not by a long shot. In the 20th century, we built tools that let us look even deeper than the cell, right into the nucleus, at the very molecules that write the code of life itself.
This image is just fantastic because it shows exactly where pathology is today. The journey of discovery didn't stop with the cell. Nope, we pushed further, to the chromosomes inside the nucleus, and then all the way down to the DNA double helix. Modern pathology is just as much about genetics and molecules as it is about looking at tissues under a microscope.
And that's where techniques like PCR come in. PCR, or polymerase chain reaction, has totally changed the game.
The best way to think about it is like a genetic copy machine. You can take one single microscopic piece of DNA, so tiny you'd never be able to analyze it otherwise, and you can Xerox it into millions, even billions of copies. This massive amplification is what lets us find the genetic fingerprint of a virus, diagnose a hereditary disease, or spot cancer markers with a level of precision that was just science fiction a few decades ago. It's incredible.
Which brings us right up to the present day. Pathology is going digital. We're talking about glass slides being replaced by high-resolution pixels on a screen. And this brings amazing new capabilities, but, as you might guess, some brand new challenges too.
So this is called telepathology, the ability for a specialist to diagnose a case from anywhere in the world. Now, there are basically two flavors. The static method is the most common. Someone takes a few pictures of the slide and emails them. Easy and cheap. The dynamic method is way cooler. The pathologist actually gets to drive a robotic microscope from hundreds of miles away, exploring the whole slide in real time as if they were right there. But this raises a really important question, especially for that common static method. What happens if the person at the microscope, the one choosing which snapshots to send, what if they accidentally miss the one tiny group of cells that holds the key to the entire diagnosis?
And this exact problem has a name. It's called sender bias. The pathologist on the other end can only make a diagnosis based on the pictures they're sent. If the key evidence isn't in those pictures, it might as well not exist. It's a completely modern problem for our digital age, right? It's like a high-tech game of telephone, but the stakes are a person's life.
All right, so after that whirlwind tour through literally centuries of discovery, what are the big ideas? What's the main takeaway from all this? So here's the bottom line. Pathology is the science of the why and the how of disease. Its history is this incredible journey from superstition all the way to molecular science, thanks to pioneers like Virchow, who taught us that sickness starts inside our own cells. And today, amazing tools like PCR are pushing the boundaries even further, while digital pathology opens up a whole new world of possibilities and problems like sender bias that we now have to solve.
And all of this leaves us with one last big idea to chew on. We've gone from looking at whole organs to tissues to cells, and now we can read the entire genetic blueprint of a disease. So, as our tools get even more powerful, where do we go from here? What do you think the next chapter